Our next speaker will be the CEO of American Gene Technologies, Jeff Galvin.
Well, thank you all for coming today. This is a really great audience, and I appreciate your interest in American Gene Technologies. So let me dive in. I have a lot of slides to cover. I'm going to talk really fast. Anybody who wants this presentation or even a fuller presentation, see any of the AGT folks in the room. Norman's standing in the back of the room with his hand up there. Just hand him a card and we'll send you an investor deck, a science deck. These are all non-confidential, so you can read them and share them with your advisors. Just let us know which ones you're interested in. I'm going to focus in here largely on HIV because we have some really amazing momentum in that, and I want to make sure you understand sort of the near-term future of AGT. But I'll start with sort of the broader picture. So we are a leading technology company in gene and cell therapy. And what do I mean by that? Well, we have focused on really the basics, the fundamentals of gene and cell therapy. So what we've tried to become is experts in how to make high probability, highly reliable, highly predictable drug development candidates for clinical development. And we've gotten quite good at that. Some of you may know us as somewhat a go-to organization for lentiviral or other viral vector work in terms of supplying some elements to what you're doing. But we're also developing our own products, and that's really our main focus. We are using largely lentiviral vectors, but we consider ourselves vector agnostic. We look at that as just the delivery layer. What's important is the software that you put on that delivery layer. And that's where our expertise lies. That's where our creativity really is. So in becoming this sort of expert in viral vectors, what was important to us was to pull together a tremendous amount of enabling and blocking IP that gave us access to a myriad of different disease solutions. In other words, if you've got fundamental things like a non-integrating lentiviral vector that allows you to shape the PK curve, well, that opens up areas that other companies can't get into without at least licensing that. But if you have redesigned genes that express at 6,000 times the normal level so that you can minimize the amount of viral vector that you need to put in, that's another thing that's going to give you advantage. If you have a process by which you can actually rid the body of any chronic viral infection by just picking a particular T-cell subset and enhancing that T-cell subset, that can be applied to a broad set of things. And then we also have already 5 granted patents in what's called the stimulation of gamma delta T cells. And what that means is that we have a way to naturally lift the normal surveillance of epithelial malignancy in your body to the level where it can eat fully formed solid tumors. I'm going to talk about that first, but I'm going to have to be very brief on it because I want to get to really the bulk of the presentation, which will be on HIV. Anyway, this $2.4 billion worth of intellectual property was done by a former Pfizer attorney who was responsible for setting the purchase price on IP assets of preclinical companies for strategic acquisition by Pfizer. So he knows his stuff, and what he did was a market comp analysis of our 7 granted patents, 3 allowed patents, 20 published patents, and all the patents that are part of our 28 patent families that I'm not allowed to talk about yet. So let's talk about first phenylketonuria. It's a monogenic disease. This is in the sweet spot of all viral vector companies. You see other companies taking other approaches to this. The 2 unique things that we've done is we have a gene that expresses phenylalanine hydroxylase at 6,000 times the normal level. And what that means is a small treatment of lentiviral vector to the liver would actually restore the normal morphology of this, of the processing of phenylalanine into downstream products by phenylalanine hydroxylase. Where are we in that project? I think we're the first ones to take the human gene and cure it in the standard mouse model. So we have good data that says that we can express the human gene at sufficient levels that we can mitigate the disease. This is a little bit older data, but we have more data in the data room, which is a little bit more confidential. But basically, the expression levels are sufficient to go ahead and make the downstream products and to reduce the phenylalanine levels, so it's within the therapeutic index of the current drug candidate to cure this disease in adults or in children, and we can do it at a very cost-effective price. It would cost about $75,000 to do it in an adult, and quite a bit less than that to do it in a child, because the liver is smaller. And one of the nice things about the lentiviral vector is that if you treat once as a child, the dose will scale. As the child grows up, the transgene is actually transferred to the daughter cells as the liver grows, and the dose scales along the way. So this is a very powerful thing about choosing lentiviruses for that. The other thing that we did is we have a patent on knocking down all 1,000 allelic mutations of the phenylalanine hydroxylase gene. Why is that important? Well, it's an allosteric tetramer that the phenylalanine hydroxylase forms, and if you have proteins in there that are misfolded sufficiently, they can misform the tetramer, and so you could actually cure the disease, and it wouldn't work because the mutated proteins that are still in that patient could actually misfold the entire tetramer. But we can get rid of all 1,000 allelic mutations in one cassette in a lentiviral vector. That's very important. Okay, onward to immuno-oncology. So in immuno-oncology, what we do is instead of modifying the immune system in order to rev it up against the cancer, like in CAR-Ts, what we do is we modify the cancer to make it infinitely attractive and infinitely stimulatory to the natural immune system. Let's see if I can quote here. So you have a tumor, and we can go ahead and inject a small amount of immunotox into it. What it does is it modifies about 20% of the cells. They start secreting a stimulatory small molecule which attracts, trains, proliferates, and activates these gamma delta T cells. These are the cells that are surveilling in your body right now, and they're the only reason that you are not rife with epithelial cancers: breast, prostate, lung, liver, colon, kidney, ovarian, head and neck, skin cancers. It's because your body is great at eliminating small malignancies, but it's bad once it grows beyond. If something escapes, you're going to need surgery and radiation, or maybe not. So we get about 20% of the cells that are causing this secretion, and these activated T cells come back and, of course, destroy the primary tumor. And what's the side effect? Well, they're circulating around your body. They're getting the metastases and the secondary tumors as well. So the bottom line of this project, what we're seeing is 85% complete remission in mouse models. We're at about 1,000 mice so far, and this is with abscopal effect. So in other words, we treat the primary tumor, and the secondary tumors and the metastases are also being removed by the gamma delta T cells without any further treatment. This is really powerful. It looks like it could work in late-stage cancers. And what's really interesting to me about this project is that it's almost like dialing back your cancer age, because gamma delta T cells don't know the difference between breast cancer, lung cancer, prostate cancer, liver cancer. So if we treat a lung tumor, and you see that lung tumor go away, and the metastases go away, and the secondary tumors go away, what's also happening is that undetected breast or prostate cancer is also going away. So it's sort of like winding back your cancer clock by, we don't know the exact amount of time now, but it might be 5 or 10 years. So we could possibly make cancer a chronic disease by you'll only need a shot to the primary tumor each time one of these things emerges, and that will do a flushing of your body, almost like a vaccine of your body. It's not a vaccine, by the way. Here's just a requisite disgusting mouse picture, but you can see that the mice over here had tumors that were more characteristic of the untreated mice. These things, when we go ahead and frozen section these, there is zero cancer left. Zero. No escape. That's very important in this, and I think it differentiates it from chemotherapeutics, of course, which, right, it's cells that survive that regrow the tumor. But it also is really separated from immuno-oncology in general, which has to be used along with other modalities in order to move the needle. Okay. We have an amazing team on immuno-oncology, and so I'm quite confident we're going to get that into a clinical trial in late 2020 or early 2021 with our collaborator, Dean Felcher, who's a director at the Stanford Medical School. Let's talk about HIV. The reason I want to talk about this is because we're submitting the IND for an HIV cure next quarter. There's so few things to do that we have exceedingly good visibility on the schedule from here on out. So IND, third quarter this year, first patient in, fourth quarter, data readouts, first quarter of next year. Hopefully at JPM, we'll be able to bring data from the first few patients that are going through this trial. So what do? What we're doing is an autologous cell therapy. We're pulling a 400 milliliter leukopak. We're isolating the HIV T cells because those are the only ones that matter. We're stimulating them so it enriches the mixture, and then we're depleting out all of the other T cells. So we're focusing on the only thing that needs an advantage in your body to protect you from HIV. So we end up modifying those by removing CCR5, the common entry point for R5 viruses, and putting siRNAs against VIF and PAT in conserved regions of all clades of HIV. If you heard of the London patient, one of the things that you'll find out is that people that got bone marrow transplants are still susceptible to CXCR4 tropic forms of the virus. That's not the case in ours because we have siRNAs that protect you from that. So this protects you against the full spectrum of HIV. So then what we do is we just culture these cells up to memory levels, somewhere between 10 million cells and 500 million cells. We infuse them, and they actually provide natural protection against HIV. If you're an HIV-infected individual, we expect that you'll be able to throw away your medicine, and you'll be functionally cured immediately. Your viral reservoir will actually be cleared by this over time, and you'll never be contagious. You can never progress to AIDS, and you can never be reinfected because you have HIV T cells that cannot be penetrated by HIV virons, so they're able to handle those virons like a cold T cell handles a cold viron. So this is just a proof that the CCR5 protection is 95%. That's all you need for R5 viruses. And it's complemented by 77% protection in CXCR4. This is what you need to give your immune system. It's an advantage over the virus, and then it's able to keep you protected. These numbers are sufficiently high to give immunity to anyone. Even if you've never had HIV, you can take this treatment and become immune for life. We're in the final stages of submitting the IND. We have a DSMB, we have a clinical protocol, we have folks that are signed up as clinical sites, and we have the who's who of HIV that are giving me the thumbs up on this program. That's why I'm so confident in this. One person who's not up there is David Pauza. He's our CSO, 35 years of experience, pioneer, and also one of the foremost globally respected experts in HIV. He's the guy who attracted folks like this, and these people are the who's who of HIV. I think we've got this one. Here's the schedule. I already mentioned it to you. Here's the executive team that's going to bring this across the finish line. We have a deep bench at the executive level. We're capable of pulling this thing off. And thank you. If anybody would like more details, please give me your card or give a card to Norman, and we'd be happy to send you a full presentation. Thank you.